Good morning. Let's have a class. We will finish talk about the staining technology. Then we'll talk about the electron microscope. Then we'll move on to the lecture 3, the slide 3. So we talk about the staining technology. And we already been practicing the lab in simple stain and gram stain. And we already been mentioned in this lecture class is acid-fast stain. Acid-fast stain using heat steaming with carbon fusion and counterstain with safranin to see a very special bacteria, which is Mycobacterium tuberculosis. We call it a TB. So these are the 3 we have already been mentioned or practiced right now. Then we're going to mention several others, which is including Indosport stain. Capsule stain, and the flagella stain. These 3, including acid-fast stain, we call it a special stain. We usually do not call it positive or negative. We'll just find the specimen if it showed up. And the gram stain, we call it a differentiated stain. Because we mentioned about it could differentiate the gram positive, which is purple color, and the gram negative, which is pink color. So that's a differentiated stain. Okay, so we want to mention several things following what we talked already. So first of all, the Indosport stain. By the way, Indosport stain we will practice in the lab on Thursday. What's the function of the Indosport stain? We want to see where the bacteria has an endospore and where is the endospore. So basically it's absence, presence of endospore, and the second thing is location of the endospore. The reason is that some of the bacteria has endospore, some of the bacteria it doesn't have. For example, there is a major 2 bacterial strains or species has endospore, which is Bacillus and Clostridium. And the Bacillus has Bacillus subtilis, cereus, Clostridium has a perfringens, difficile, all catenins, all those different categories. We'll mention in the examination too when we move to the disease part. So just let you know, Bacillus and the Clostridium are 2 major species has endospore. Now the location of the endospore is genetic dependent. So what that means, based on the location of the endospore, we maybe can give some estimation of what kind of the bacteria it is. So for example, the location of Bacillus, that could be central. And a location of a Clostridium, that could be subterminal. And the location of some of the other bacteria, that could be terminal. So we're talking about the purpose of the Indosport stain, then we're going to talk about how we're going to do it. Basically, the smear preparation is the same. And we talked about this already, air dry heat fixing. But the major thing here for the Indosport stain is very similar to the acid-fast stain because Indosport is a rigid system. And it's a very strong covered area on the outside, and its basic dye cannot go inside. Therefore, we need to use a very special dye called the malachite green. At the same time, we have to add heat steaming for 5 minutes. And then we also have to do is a counter stain with methylene blue. So at the end of the day, what you will see, very possible is you looks like something a little bit of blue, a little bit of greenish color. That is possible is an Indosport. And then you will see a bunch of the other area which is a very light blue. That is a background. And we will testing this in the lab using Bacillus cereus as an example, or Bacillus subtilis. Now there is one thing I want to mention, because Indosport, the basic function is to prevent the bacteria in the stress environment, against the stress environment, like some UV lights, a very cold, a very hot situation. So the Indosport stain using the bacteria culture needs to be old. This is a major difference compared to the gram stain. And we mentioned the gram stain is focused on the cell wall structure. So we need to make sure the peptidoglycan is complete. If the peptidoglycan is already cracking down, let's say you're using 72-hour-old culture, then all it's going to stain gram negative. We call it is gram variable stain. And we did this in the lab also. However, for the Indosport, we want the bacteria to form the Indosport. Therefore, this needs to use very old culture. Now, how old it is? At least 72-hour-old. Most of the time in the lab, we were growing about a week. Okay, a week-old bacterial culture of the Bacillus subtilis was showing a very good Indosport. Because the young culture is still developing the Indosport. And we will mention again on Thursday when we move to the steps, we talk about specifically for bacterial Indosport. Okay, so that's the Indosport stain. Next one, capsule stain. So first of all, what does a bacterial capsule looks like? If you look at the picture, there is a very typical example, which is Streptococcus pneumoniae. And the Streptococcus pneumoniae, you could see a very clear, inside the two dots there, it's diprococcyl. Diprococcyl. And they covered with a very heavy smear. This is one is a polysaccharides. Now the function of the capsule is actually, we call it an anti-phagosome. So the story is that when a bacteria coming, let's say that the bacteria coming in your body or in the animal's body, most likely at the beginning something will be engulf or grab it. This is called a macrophage. And macrophage. The macrophage is one component of your white blood cell. And when you grab it, they will do a lysis. The lysis usually rely on the enzyme, it's called a lysin. And then you actually is cracking down the peptidoglycan, the cell wall structure. So that heavy capsule, which is called is help to prevent the bacteria attacking by the macrophage. Well, by the way, this is the first defense system of the body before they move on to the real immune system. So the capsule is important for the bacteria to survive. So how to do the capsule stain? There is two methods to do. Number one, we already mentioned it. We can do a negative stain. So we actually not staining the bacterial capsule, we're staining the background. And you can see right there on the top, the background is purple and the diprococcyl showed up. Now what type of the dye we can using? It is Indian ink and nigrosin. That can be using to do the negative stain, staining background. Now the second method, what we can do is we can directly stain capsule. We're using carbon sulfide. It will be staining somehow like a greenish color, and we'll show in the capsule. Okay, so there is two methods to do. Directly stain using carbon sulfide, or you do negative stain using nigrosin or India ink. So that's for capsule stain. The last one we want to mention is a flagella stain. So first of all, bacteria has flagella. It's usually like a tail. Well, it's a different flagella, a different type of the flagella, monotrichos, amphitrichos, lophotrichos, and the petrotrichos. We'll mention about next section. Now, the flagella, usually at the end, is generated by a protein called flagellin for the bacteria cell. Now, for the fungi, what it looks like, looks like a circle. 1, 2, 3, 4, 5, 6, 7, 8, 9, and in the middle there is a 2. So we call it a 9 plus 2 configuration for a fungi. Because the flagella composed by flagellin, it's a protein, so it's very soft. So we cannot just do a gram stain. The reason is if we're using the gram stain methods, it's very easy to be melted. So how we do? We need 2 glass slides. One is about 45-degree angle, and the other one is just lay down on the bench. And very similar, like you did for the blood cell calculation. So how you do? You need to let it roll. The flagella is very thin, very soft, so let it be rolling down. It could be rolls, goes here, okay? Once it rolls, we have to, first of all, fix it. So we have to do some fixing. What are we going to do fixing? Formalin. So use formalin to do the fixing. And then we might need to make sure it is coated there. So we add tannic acids to do the coating. So tannic acids needs to be there to coat it. And the last, we need to put some dye. So we add basic fusion. Like carbon fusion, you can put it there. Okay, so later on, what you're going to see the flagella is very thick, like that. So the flagella stain will be showing up. Now, why the flagella is important? Because flagella stain certainly tells us the bacteria have a very strong moving capability, because it's a real move, not like a vibrate. And you can see a very typical example for the flagella stain is this one. And another bacteria we're going to mention in the lab all the time is this guy, is Proteus vulgarius. The Proteus vulgarius, this pathogen, originally from soil, what happens, the flagella is covered the whole surface. So what's going to happen is this bacteria cannot form in colony on the plates. It will form a large smear at a long area. We usually call it a swarm. Instead of you have a colony, they will not form in the colony. Okay? So this is here, we finished talk about the staining technology, and because of the time and the lab facility, we can do simple stain, gram stain, Indosport stain, and acid-fast stain. I do not have a microbacteria anymore, so we skip that, but originally we could do that one. But for the others, we're not going to do it, because its condition is not that good, so we didn't do it. Here we have a slide. I think it's a very nice slide. You can see the gram stain. And the gram stain here, you see the first one is very similar to Staphylococcus aureus. And I did it in the first lab. It's really nice. Second one is acid fast stain. You will see it's for tuberculosis. Then Indosport stain. This slide is a little bit confusing. Usually we see it's a little bit greenish, not that pink color. Capsule stain. This is actually is you stain in the background. Last one, flagella stain. You see the flagella is showing up. Okay, so very nice. These 5 pictures really showing you the different color of the staining technology, what it's going to be looks like. Okay, next one, we want to talk about, spending some time, talk about electron microscope. Now, to well understand the electron microscope, we want to do a comparison between light microscope, then you have a fully understand what's the difference between that. Instead of we are just talking about the electron stand, the electron microscope separately. So we want to draw at the top. This is light microscope. And on here we're going to do electron microscope. And we will using the example is TEM, which means transmission electron microscope, which means we can see the internal structure. And the scanning electron microscope, we will mention briefly, because that's only on the surface to see the 3D images. So if you still remember in the lab what the light microscope looks like. We have a lens. That's not a problem. Objective lens. Then we have a stage. So you have a light resources right here. That's a light. You have a condenser. And on the top, you have 2 things come up here, is ocular lens. Okay, so we draw ocular lens. This is objective lens. And this is a condenser. And this is a light. And what is the light comes from? The light ray comes like this. Some refracted, some directly going through. And your eye is right here. Okay, to see it. Let's say your eye. Okay, now, what does the electron microscope looks like? So first thing first, the energy resource. Light microscope, energy resource is light. And I said, plug in, turn it on, all the way to the bottom, which means the light is the biggest one. Electron microscope, it is something not light. Some here, like a bullets, like a gun. So what is called? It is electron beam. So, light microscope, you have those light rays come out. An electron microscope, you're going to come out with what? Electron rays. That's easy understanding. That's the first thing. Okay, then the second thing is type of lens. Did you notice that your condenser, your objective lens and your ocular lens is all glass? And I said it's very expensive, don't block it. Then how about the electron microscope? It looks like a circle One is here, second one right here, another one is on the top. And people make a joke for your understanding. These looks very similar to a donuts. Like you buy donuts at Dunkin' Donuts. So donuts like electron magnets. So instead of you have a glass, here what you have is an electron magnets. Now, of course, the light, here you have a light ray, and this is an electron ray. Okay? So that's some major difference. Now, we want to talk about the medium. Light field microscope, we talk about light reflection. So, you have a glass. What's the middle between that? Is air. So it's easy to understand, it's air. How about the electron microscope? This whole thing needs to be a high tunnel vacuum. This needs to be a vacuum, not air. Okay, that's important. Adjustment. How we do the adjustment of a light field microscope? Remember, we have a focus, which is coarse focus, fine focus. So we use coarse or fine focus, which means mechanical or manually, we say it. You just twist it. How about the electron microscope? You have to adjust the electron current. You adjust the electron current, it will be changed. So at the end of the day, instead of you using an eye to see it, on the top you will see a screen. Everything is showed up in the screen. That is important. Okay. This is you use the eye to do the observation. This is you showing up in a screen. Okay, now something we really interesting. What is the magnification? How enlargement could be a light field microscope we mentioned? 10,000, is that right? So we say 10,000 to 1,000, sorry, to 1,500 x. How about the electron microscope? If it's transmission electron microscope, could be around 100,000 x. Now why? This is a light microscope. Light is an energy resource. The lambda is around 625 nanometer. This guy is the electron beam. This lambda is about 0.2 nanometer. And the smaller the better. Remember the resolution power? Now we're going to talk about the resolution power. Lambda divided by 2 multiplied by Na. The same thing. So what is the resolution power of a light microscope? Is that 260 nanometer equals 0.26 micrometer? How about the electron microscope? Very small. 0.02 nanometer could be. That's why you could see a virus very clearly. Okay, last thing we want to mention. How we prepare the sample. Smear preparation, is that right? Smear preparation. As as we talk about, and then we do a staining technology. But how about the electron microscope? It's not the same thing. It's a step a little bit complicated. We call it a freezing etching process. So usually what they have are multiple steps. Number one, the sample you have to do dehydration. Take out of all the water. And the second thing is that we have to do a very fast freeze drying. How we do the freeze drying is liquid nitrogen. To do a deep freeze drying. When the freeze drying happens, what are we going to do? We have to slice it, we have to cut it. So cut into very thin piece. And this moment we call it a shadow. The shadow going to generate. And then we have to do a coating. The coated with heavy metal. The heavy metal coating usually is a lead or sometimes is a platinum. So therefore, what it looks like, the inside you can see these various interesting material inside. You have see the inclusion, which means the storage of like a phosphates, magnesium, or those chemical or nutrition ingredients there, and you will see the different structure inside because we can do this shadowing. And it's a freeze drying. You have to cut into very small pieces, and then you put into the tunnel. And it's all adjusted by the electron current and showing on the screen. So this is a transmission electron microscope. And you can see the slides right there, and this is almost a complete table to see the difference between these. And electron microscope, most of the time, we cannot just naturally do it. WVU across a street, there is an engineering department, have a shared facility, have some electron microscope, and you need to get the training first to get the certifications and can do in your research. One of my graduate students using the electron microscope to see the stoma of the leafy green, because we try to see the waxy amount to affect the salmonella grow on the different leafy greens. So that's why we want to see the stoma infected by those type of the wax. So that's why we use an electron microscope to see the internal structure. But not everybody can do. You need to first of all do the training, and especially if you want to do the pathogens, and there is a more strict training for that. Okay, so just want to let you know. Now I want to be very briefly talk about the scanning electron microscope. Scanning electron microscope. You basically is on the surface, you do like a back and forth scan. And the signal will be transferred to a photo amplifier. Sometimes we call it a multiplication multiplier. And then on the screen, you will showing those 3D images. And it's very similar to the laser confocal microscope. And we want to see the surface. This is what it looks like. And this is also another example. You can see what the scanning electron microscope looks like, but not the inside. Not the inside like cell wall, nuclear, ribosome, all those things. So, scanning electron microscope, the magnification usually is about 10,000. Some cheaper one even 5,000 to 7,000. So I will say they are a little bit higher than the light microscope, but not as that big as the transmission electron microscope, because we certainly did not require them to see the internal is on the surface, which is give you overview of the microorganism, what it looks like. So certainly it's a lower requirements compared to you want to see the internal structure. Okay, so that's the description of the comparison between light field microscope and the electron microscope. Now, last thing, we will be just using the slides to talk about shape of the bacteria. The shape of the bacteria usually means morphology. That is what we care about. So we mentioned in the lab, it could be coxal, it could be those very short straight lines there, we call it rods. It could be curves, vibro. It also could be something a little bit large, but not that large, we call it a coccobacillus. It could be also like this, it's filaments, like a spiral sheets. So that's the morphology, which means the shape of the bacteria. Now the second thing important is the arrangement. Okay, look at the picture here. The arrangement is very important because somehow it tells you what kind of the bacteria it is. And we mentioned about leprococcale. It's very typical Streptococcus pneumoniae. Because this is the #1 community-acquired ammonia in the United States. We'll talk about that later on. It could be 4 of them together, tetrad. The bacteria we try to do in the lab, which is Micrococcus ladeus, which is actually a tetrad. And I try to activate it to see whether it can grow. Okay? There is some other bacteria, could be 8 of them stacked together. Very few, not too much. We call it a succini. And more often what we see is a bacteria like Staphylococcus aureus is a grape-shaped cluster. So very interesting. We will mention again, if there is a young lady who has a sexual activity life, didn't go home very often, spending the nights outside a couple of days, when they have a urinary tract infection, then the doctor could be isolated some bacteria, some Staphylococcus from the vagina area or from the urinary sample. There could be Staphylococcus agalactia, which looks like that. That is the #1 sexually transmitted disease pathogen for the mid-year or young ladies, which has a very prolific sexual activity. It's specific for the causing a urinary tract infection. So something, the example it tells you is shape arrangements tells you very quick what type of the pathogen it is based on the symptoms. And another example, Staphylococcus aureus, and I mentioned the chicken salads. If you have a chicken salad, 6 hours later you have a vomiting diarrhea, very possible contaminated with Staphylococcus aureus. And what it looks like, you will see in the lab more clear, gram-positive, grape shape. Now here are the other things. Vibrio chloride, usually jellyfish will happen. That's vibrio. It happens in the jellyfish a lot. In the deep ocean marine area, the jellyfish contaminated with vibrio. And if you eat it, what's going to happen? Higher amount of the water going to lose the dehydration. So what happened, how severe it is, 6 hours, you lose 10 liter water. So if you do the treatment of the vibrio, the first thing is not antimicrobials, is actually rehydration. That's what they do. Campylobacter jejuni, spiral shape, Campylobacter. This is a pathogen we will mention in our exam 4. Campylobacter jejuni. This is a very interesting bacteria. It is a microaerophilic pathogen. which means they don't like oxygen too much. They require to grow only 5% oxygen, about 15% carbon dioxide. If you have more oxygen, it will be died. It's a number 2 pathogen for the poultry meat. And very ironically, it is very difficult to grow in the lab. However, it's a big risk for the poultry industry. Let's Campylobacter jejuni. This one we mentioned about the hook shape, Leptospiro interrogans, if you remember. That's belonging to the spiral sheets. It's a hook shape. Leptospiro interrogan. So what we have to do, we're using dark field microscope to do the observation. And that what is showed up with the dark field microscope. The background is dark and the specimen showed up. Last one, Bacillus. Bacillus is large rods compared to the E. coli. E. coli is a tiny rods. You see a couple of times already. It's a special simple stem. Bacillus is large rods like a chain. We will do the Indosport stem, and you'll see it more clearly on Thursday. Okay? This tells you the shape and arrangement of the bacteria is very important. Therefore, I always make sure you're understanding for the gram stem, there are the 3 major items you have to record. It is gram reaction. This is everybody knows, positive or negative. But you may be going to forget about describe morphology. And the more important is arrangement. So these are the 3 major items to record a gram stem results. Gram reaction, morphology, and arrangement. So make sure in your lab notebook, you have those 3 important information after you're done for the gram stem. Not only the gram positive or gram negative. Okay? So that's important because you see all these examples, the shape and arrangement, very typical corresponding to most of these passages. Okay, so we finished here. We finished exam 1, lecture 2, which is the slides. So we're gonna move on to the lecture 3. We get a bunch of time, we're gonna talk about these, okay? It's a very interesting topic and lots of information here. We want to talk about that. Something you already know in the other classes, we maybe just make sure it's short and easy. Some other things we will be emphasized. So what will be the lecture 3 talks about? This will give you an overview of the composition of a bacterial cell, or we say a microorganism cell, but typically we're using E. coli as an example. It's going to relate it to some of the chemistry or biochemistry knowledge. We're not required to know every single chemicals, but I will introduce you in the lecture, and some of them will be required to remember later on, okay, not at this moment. First thing first, what's the major difference between prokaryotes and the eukaryotes? So let's say I have an E. coli, and I have a fungi, let's just say. Prokaryotes versus eukaryotes, what is the major difference? A couple of years ago, I have a question in the exam. So one of the students said, prokaryotes is simple and eukaryotes is complicated. It looks like that when you see the slides, but it's not really true. I will explain to you. You look at the E. coli, there are only so many items there. However, they have to support their whole lifetime. So which does not mean it's simple. It's maybe more complicated than a eukaryotes, because eukaryotes, each of them are functional individually. Therefore, what's the major difference? It's a membrane bundle system. Bundling system, is that right? So we're going to be very briefly tells you, prokaryotes, we say it's naked. everything stuck together. No cell membrane to separate them. Fungi, eukaryotes, is more individual function. There is one theory, one hypothesis to describe that is called endocytosis. Is that right? So they think about a long time ago, fungi maybe eat a bacteria. So the bacteria, the genome sequencing has been integrated into the chromosome of the fungi, and later on, the genotype become a phenotype, so they developed all these individual organelle, or we say cell machinery. So could be lysosome come from a bacteria. It could be Golgi apparatus come from another bacteria. They eat it and then they integrate it, they become friends and they stay there forever. Looks like a parasite. That's one of the theory called endocytosis. Okay, we will jump these. We're not going to talk about that. And the details of the eukaryotes you learned in your biochemistry class, plant science class, animal science class, or the other classes, or biology. So we're going to skip that. We're going to focus talk about the bacteria. Whichever you say, it is a little bit simple. Okay, now, the very simple question for you. If you don't study this before, let's think about what the inside of the bacteria cell. Okay, let's say I have a bacteria. I have an E. coli. What we have? Obviously, you need to have a genetic information, something. So what inside? You need a DNA and you need RNA. What's the name of those? Nuclear site or nucleotides. Okay, they are there. What others we need? Look at the structure there. Lots of the components are protein. Is that right? Which place to biosynthesize protein? Large unit, small unit. What is called? Ribosome. We need a ribosome. I'll call it 50S, 30S, then add it up together is 70S. Why is not 80S? Because it's a Vandenberg unit. It's not simple plus or minus. Okay, so the ribosome we need. Is that simple? For bacteria, we need some places to storage some of the nutrition ingredients. So somehow, they may have something right there. You can see it. Something like this. We call it inclusion. We mentioned for storage nutrition. So that's inside. Pretty simple. Like some people answer the question. What's outside? First layer, cell membrane system. Or we say plasma membrane, different description. Now what's the key for a bacterial cell? Always remember, what it will be the major difference between a bacterial cell to a fungi to the human being, which is outside here, which is actually a cell wall. This is the key. Because of bacteria have cell wall, therefore we can develop antibiotics. Because antibiotics will target the cell wall. Do we have cell wall? No. That target is specific for bacteria. Therefore, ideally for human being, it's safe. Always remember, plant cells and the bacteria has cell wall. Animal and the human being do not have the cell wall. That's a major advantage of the bacterial cell. Relate to the treatments. Now what are the outside have? Now sometimes we have a capsule for the protection. Okay? Now what else we have? We may have a tail right here, going to have to do the movement. So that's a flagella. Now, very important for E. coli. Remember E. coli 0157H7. What did this H mean? It is a flagella antigen. Because of this flagella, we could be serologically typing, give them an ID to recognize it. If they do not have a flagella, you may not can identify it somehow. Okay, so flagella is important. What else we have? Sometimes we have a little bit of these extruded material. We call it a pili, or we call it a fimbrial. Now, what's the function of the pili? Genetic communication. What are the major three genetic communication methods you learned in your other classes, if you still remember? Transformation, transduction, and conjugation. And the pili is to using for genetic communication during the conjugation process. So some people say a little bit motility function, but more important is genetic communication. This is well explained, the picture right here. So if you draw a picture by yourself, you're going to understand it. And this slide, which tells you a basic function and an overview of a prokaryote cell structure. Cell membrane, the barrier, cell boundary, nutrient transport, metabolic process, and detect the signal. Response to the stress for the bacteria. Cell wall, its protection, provide the shape. Ribosome, protein synthesis. Capsule, resistant to phagosome. Pili is for attachments, conjugation. Flagella, motility, and inclusions for storage, nutrition ingredients. So basically, when you draw this picture here, you respond to all the information in the slides. And this is a way what we should learn is have ample